All right. Hi, everybody. We're going to get started. This is Jay Scott. I am co-executive director at Alex's Lemonade Stand Foundation, and I'm pleased to welcome you today to the latest lecture in our childhood cancer lecture series, which is Patient-Derived Models of Childhood Cancer. And our presenter is Pat Reynolds. A little bit about Pat. Pat received his BA in biology from University of Texas at Austin and his MD from UT Southwestern in Dallas, and his PhD from UT Austin. His pediatrics trainings was done at National Naval Medical Center, and his postdoctoral fellowship was in cancer immunology, also at UT Southwestern. Interestingly, Pat holds the rank of Commander in U.S. Navy, where he is retired. He is currently the Cancer Center Director for the School of Medicine at Texas Tech University, and he's also the Director of the South Plains Oncology Consortium and the ALSF COG Childhood Cancer Repository. We're really looking forward to Pat's lecture today, and with that, I will turn it over to you, Pat.
Thank you very much, Jay, and thank you all for having me and letting me talk about one of my favorite topics, which is models for studying childhood cancer. We're going to talk about primarily patient-derived models, but I'm going to mention all of them briefly for you all. Of course, everyone's familiar with these, I believe, but we'll go over them. Cell lines, and then the newer version of these organoids, which are cells growing continuously in culture. They can come from either human patients or animal cancers. Patient-derived xenografts, which are human cancers grown directly into immunocompromised mice, so you don't go through the selection pressure of going in vitro first. And then genetically engineered models. And we're not going to talk about GEMs today, but I do want to mention them. Germline alterations that give rise to a given cancer can be introduced into the germline of mice and generate in de novo inappropriate organs occurring in the mice. And the advantages are that they have these tumors occurring usually in the spot where they're going to occur and with intact immune systems. But there are also disadvantages to these. One is the artificial ways of driving the oncogenesis that's usually used. The fact that these animals will come up with cancers at various times and making experiments difficult. And in particular, in terms of studying drugs, is the lack of exposure to standard therapy. So these are like chemo-naive patients, if you will. And so these disadvantages may outweigh some of the advantages. There is a lot of work that's being done with these. Whether clinical translation is going to come out of these still remains to be seen. And they're certainly interesting models. But what we're going to focus on is really what we get directly out of the patients today. And so these patient-derived models, first of all, traditional cell cultures and in the form of cell lines, these growing continuously in culture, either on substrates or in the case of lymphomas and leukemias, and in fact, neuroblastomas to some degree will do this a lot, growing in suspension. And then organoids, the more modern, if you will, version of this, where the cell cultures are generally grown in clusters and they're growing in suspension rather than on a substrate. And there's a lot of thought that these may be better than cell lines. The data has yet to be actually materializing. And in fact, if you look at the cultures of childhood cancers, they often grow as essentially organoids. So there's a continuum across here. And so we're not going to spend a lot of time on this. I will say that there are some people that have gotten samples from the repository working on these. And this is an interesting approach. But what we're going to really talk about, everything we show you today, is really more traditional cell lines. But as I said, the way they typically grow are basically a hybrid between those and organoids. And then there's patient-derived xenografts, which are the cancers grown directly in immunocompromised mice. So generally subcutaneous. You'll see some people doing orthotopic xenografts. Whether or not those are an advantage has yet to be demonstrated. For the leukemias and lymphomas, you'll often see this as being intravenous, and what they do is the lymphoid ones in particular will populate the spleen, and that's where they can be harvested from. But the cells will circulate across the mouse in total. So an important part of these relative to other types of tissue that you might get, i.e. patient tissue itself, is that all of these are renewable resources. So they can be provided to many laboratories. Laboratories can replicate exactly what was done in others. And of course, they're live cells, and so you can actually study drugs on them. So that's one of the advantages of these. Now, of course, the disadvantage is we're taking them out of the patient and putting them either into cell culture or into these mice. So the real challenge here is to figure out how we make these cell cultures and these cancers growing in the mice as close to what's going on in the patient as possible, so that we can use them to understand actual human cancer. Now, I feel very fortunate that ALSF supports this repository that is the Children's Oncology Group repository for establishing and banking cell lines and xenografts. And this is a centralized resource lab that receives samples from across the COG. And as you'll see, we place them into both culture and directly into patient-derived xenografts. And it's really turned into a worldwide repository for childhood cancer in that we provide about 600 labs, more than 600 labs in 28 countries with these models. And as I mentioned, it's generously funded by Alex's Lemonade Stand Foundation, so that people requesting these models can simply provide a FedEx number or something that covers shipping, and there's no other costs involved for them to get the models from this from the laboratory into their laboratories. This is our website. And as I mentioned to Jay a little bit ago, we have a large amount of models that are about to go up on the website, a big update that's coming. So within a month or two, come back here and take a look at this. There's going to be a lot more there. In the meantime, if you have needs, you can go there and look at the models we do have. And if they don't meet your needs, just contact us via email. And we can look at the data that we have and provide you models that are more appropriate for what you need than what's up there. And I just thought I'd show you the international use of this. These are the top 16 countries now using the repository, of course, mostly in the USA, but we do have quite a bit of users across these countries as well. Now, I want to pay some attention to quality control and regulatory issues on this. First of all, it's important to note that all patient materials obtained via informed consent and that patient confidentiality is strictly preserved. And we're able to annotate these because most of these patients that we get are on COG studies of some sort. And so we can use the COG patient code number to link back, and COG provides us some clinical data that way on these. We don't have it, the COG does. And that allows us to fully annotate these with actual patient information and still preserve the confidentiality of the patient that provides us with the sample. This is important also because it's not uncommon that we'll have people contacting us with a journal request that we provide them a note that all patient material was obtained via informed consent so they can publish their data. So that's an important component of it. And another aspect of this is we're very careful to make sure that what we're giving you is what you think you're getting and what we think, and that it came from the patient we think it came from. So every single one of these models in there is validated to patient origin. For the older ones, we don't have actual patient material, but for all the new models, and there's a large number of them, we actually have original patient material that we've run DNA short tandem repeat assay on, and those are used to compare and make sure that what we're giving out is coming from the patient we thought it was. We also test them to make sure they're free of microbial contamination, they don't have any mycoplasma, and that they're not EBV transformed. And this is important because, especially with the older patients, we'll get some of these that grow out that are really EBV transformed lymphoblastoid cells, not the tumor itself. And this does occur in PDXs, not just in cell lines, but it's mostly a cell line problem. All the PDXs are tested and validated to be free of human pathogens. We do this by an outside laboratory, so we can just provide that without having to worry about the liability of it, and they're a test lab. And this is needed for many of the animal facilities will require this if you're going to put human cells in the immunocompromised mice. So those data are available for all the PDXs that we distribute. And the subsets of cell lines and PDXs, as you'll see, some of them receive some very extensive genomic characterization. As we get more models into various diseases, we're hoping to have a lot of these data going forward. Right now, most of this, as you'll see, is in neuroblastoma. We also, and again, mostly in neuroblastoma right now, we hope to extend this, is to determine responsiveness to standard therapies so that you can have those data to work with and compare to the genomic characterization data. Now, for the cell cultures, it's really important to spend a little time about cell culture medium. First of all, people need to realize that these bicarbonate-based media that we all use are going to change pH a great deal over time, and especially with cell density. And this is a remarkable change in pH. So this can be diminished somewhat with using things like Heppy's buffer. And so you need to pay attention to your cell density, your time and culture, and the media you use, because pH is going to confound some of your results here. A second problem we find that people don't realize sometimes, that fetal bovine serum varies a great deal with its ability to support growth of cells in vitro from lot to lot. For the repository, we scan through a lot of very large lots, and we pick one that's ideal. So if you're having trouble growing cells, this may be one of the issues that you're getting into. I just got a difference on my screen, Jay. Is everything still going through okay?
Yeah, everything's going through okay. It's just it's in presenter mode, so we see your next slide over to the right, which is fine. It's not a big deal. But if you don't want that to show, you could just take it out of presenter mode.
Okay, yeah, thanks. Well, actually, what I should do, I guess, is I should have done that originally, is just switch the slides, the screens. But if you can you see them okay? I'll just leave it like that if you can.
Yeah, we can see it okay.
Yeah,
we just get a preview of what you're going to talk about next.
That's fine. We'll just leave it like that then.
Okay.
I should have switched the screen. Sorry about that. Some of the established cell lines, as I said, will only grow, are not going to grow in the fetal bovine serum that you'll just get. You've got to make sure it's good fetal bovine serum. And some of them, and we annotate those in the repository, are only going to grow in serum-free media. We use neural basal medium for some of the neural cells and neuroblastoma, the gliomas, etc. Some of them will only grow in this, but most of them will grow in fetal bovine serum containing medium. One of the other things that I wanted to mention here is this growing in pen strep. People typically do that, but you've got to realize that's going to mask mycoplasma. So everything we do in the repository is antibiotic free. We put the original clinical samples into gentamicin at the outset for a week or two, just to make, because nothing coming out of OR is absolutely sterile. But once we get done with that, we go antibiotic free so that we can not be masking this mycoplasma. It forces people to be very careful with their culture conditions. And so what you're getting is antibiotic free. And we encourage people to go to that for your cell culture because pen strep does change cells actually, but most importantly, it'll mask this mycoplasma, and that you'll pick it up right away if you're antibiotic free. If you're not, it can be there and you won't see it. And then you've got that contaminating your results. And finally, trypsin that people like to use can carry mycoplasma. We don't use it at all. We use EDTA. Most all cell cultures can be passed with just EDTA-based divalent cation-free salt solutions such as puck-saline. And we recommend doing that rather than using trypsin. And let me just pause a second here, since we had that business with the screens. Let me see if I can just switch these here. Actually, I'm sorry, but I'm not sure how we switch screens on this. So I'll just go back to sharing here, and we'll go with this. So another issue that I want to cover is cell line and PDX cross-contamination. And people have now paying attention to this. For years they won't. But I can't tell you how many times we found that problems exist because people were working with what they thought they were, and they weren't working with what they thought they were. A single cell getting in will outgrow the other cells in a culture. And so you have to be very meticulous. And even with meticulous cell culture procedures, even in labs with all the rules in place, you still get some cross-contamination here. People mislabel flasks, or cells get splashed over. You never handle two cell lines in the hood at the same time, but it still happens. All right, and so it's really essential to monitor the cell lines and the PDXs with short tandem repeat profiling. It's not very expensive. Many labs will do it for you, and this is something you want to monitor. These short tandem repeat profiles are available in a searchable database. So if you have something that doesn't look right, you can search and find out what you actually got. And if it's a mixture, you may have to work with people to sort out what's there. But if it's just one line has taken over for the other, you'll be able to find out what you're working with by just searching in the repository website. And it's important to note that PDXs can be mixed up as well. You see the animals get mixed up by the animal handlers when they're changing cages and you don't know about it. So you've got to monitor this in PDXs as well. And finally, mouse lymphomas will occur and in these immunocompromised mice, and they can replace your human cells. Doing short tandem repeat profiling will pick this up. You'll see these that you're not getting a human signal. And there are also PCR techniques that you can use to look at human-mouse ratios, which will also useful to know what's going on in your PDXs, but that'll quickly pick this up as well. Now, when we get in samples, here's what we do. We establish these into culture in 3 different culture conditions. Yes.
So I'm sorry to interrupt you, but we're only seeing the intro slide now.
Oh.
We're seeing the intro slide, but then all your other slides along the left that are small.
Yeah, I'm not sure what I did when I went back to that, and now I'm not sure. Let me stop.
So you could go back to the presenter way. I think that was fine.
Resume share.
Okay, now we're seeing establishing cell lines and PDXs, so it's slide 10.
So let me see if I can, do you know how to switch to, there's a way to switch these screens and I'm not too sure what it is.
Someone said at the bottom, maybe where it says share screen,
Yeah, I've got
or of the PowerPoint. Click full view.
What are you seeing now?
Now we see the presenter mode.
All right, so you can see the slide I'm looking at, right?
Yep, yep.
Okay, so, well, let's go with this. You can see that, that's fine.
Okay.
So in establishing these lines, we put them into 3 different culture conditions. And these were chosen to replicate what people standardly do. And we're going to go in this in detail in a minute, this 20% oxygen. The 5% oxygen, which is really bone marrow. And if we're talking with leukemia, lymphoma, or metastatic neuroblastomas, often they come from bone marrow. That's why we chose this. And then trying to replicate about what's in tumors. And it varies a large amount across the tumors, but just picking 2% as a 10-fold less than 20%, this is what we went with. And you'll see there are some differences in this. But these 3 culture conditions, you'll see, have told us a lot about what people that are studying just these cells are getting. And it's only a fraction of what's out there in the patient. We also put these directly into the mice, so and these are NSG mice that we put them into, so that they can be grown into nudes. Some of them won't, but most of them can be passed into nudes afterwards. But we use that because of the low lymphoma rate in the NSGs and the lack of NK cells and the better ability to grow these cells, particularly if you're talking about the lymphoid malignancies. And then we cryopreserve from anything that grows out of this. Cell lines can be, as you know, passaged into mice as cell line-derived xenografts. We use those just like everyone uses that for some experiments. But for generating the repository, what we're generating is these 3 different culture conditions and the patient-derived xenografts directly into the NSG mice. And Jay, just to check, you're still seeing the slide advance?
Yes.
Great. So oxygen tension in the cell culture, and why are we doing this? Well, traditional cell culture in room air is typically taking the room air plus 5% CO2, which is close to 20% O2, and there's nothing like this in the body. The well-oxygenated tissues are around 8% oxygenated in the body. Bone marrow, as I mentioned, is 4 to 5%, and not only is that's in the older literature, but we actually, when I was at Children's Hospital Los Angeles, we actually got the RB approval to check this out, and we found that this is true in children as well. The older literature was in adults. So it's really down around 4 to 5%, and that's in non-metastatic involved marrow. It's probably a lot lower in the marrows that have a lot of tumor in them. And then the tumors are somewhere below 2%, depending on where you look in the literature for this. We haven't been able to do that effectively in patients, but we have gone in with oxygen probes in the PDXs and they are hovering down below 1%. So we're talking about a very low oxygen condition relative to what people are growing when they're doing standard culture conditions. So this is one reason why we paid attention to this. Also, Mike Sheard had worked with us a number of years ago and looked at this when we were looking at acute lymphoblastic leukemia samples, and found that they really wouldn't grow at 20% O2, but they grew very nicely at bone marrow level oxygen. Not a big surprise. They're going from the bone marrow right into bone marrow level oxygen. So again, this diminishes the ability to grow cells. And you'll see this, I'm going to show you some data in a minute. So when we're going to look at this in neuroblastoma, because that's where we have the most amount of data, and you'll see that just like the leukemias, a lot of these won't grow in culture conditions that many of you are using as your standard culture conditions. Look at this table here, and here's a larger number of samples, but out of those larger number of samples, a total of 162 grew in culture. And when we looked at, and these are not all the samples we've ever tried to culture, but these are the ones where we tried all three conditions, 2%, 5%, and 20% on all of these. And a number of these also went into patient-derived xenografts and grew out as PDXs. But if you look at this, those that'll grow in all conditions are roughly about half of these, okay, will grow in all conditions, so they're agnostic to the oxygen level. But look at here at those in 20% only. In other words, they'll only grow in the culture conditions that everyone uses. Only 4 of them would only grow in 20% only, okay? In those that would only grow in hypoxia, whether it's 2 and 5% only, 32 of them. Ones that would grow in only 5%, 33 of them. So, and some of them would only grow in 2%. So you're looking at about half of these cultures that we got in neuroblastomas that would only grow out if we use some sort of hypoxia. They would not grow at all in the 20%. And some of these will grow in 5% and 20% only, okay? Not surprising, there's just none of them that grow in only in 2% and 20% only, because that's such a wide variation in oxygen concentration. So when you're working with 20% only cultures, you're working with only a fraction of what's coming in from the patients. And so keep that in mind and really think about getting hypoxic culture conditions going in your laboratory, because that allows you to look at the other half of the patient-derived models that won't grow in these conditions that everyone uses. Now, let's look at another impact on the ability to grow these things in culture. Again, this is neuroblastoma data, where we have the largest amount of data. And that's looking at the phase of disease and the site of the sample, okay? So first, in cell lines. If we look at, we get some bloods, and there's a lot of metastatic patient tumors in blood at diagnosis of neuroblastoma, so not surprising we can grow some of these, okay? As we'll talk about in a little bit more detail, we have focused on trying to get post-mortem samples going, because these are the samples that reflect the tumors that we failed on, that we cannot stop from killing the patient. And we found that we could grow these most successfully out of blood, because the post-mortem, the oxygenation is maintained there. There's a lot of circulating tumor. We do this not just for neuroblastomas, we've done it for rhabdos, Ewing's, various other types of tumors that are circulating. And look at this, you can get 78% of these to grow out of the blood when they're post-mortem. So a huge difference between diagnosis and of course, very late disease. And you see this across the board here. First, looking at just the sample source, most of what we get for neuroblastoma is bone marrows. And at diagnosis, you can grow about 20% of those. You can grow about 15% of them at progression. So not a big difference. Same true for tumor. Not a big, you get more to grow out of tumor because you get more sample to work with. But with the progressive disease samples, not a whole big increase in those. And we don't get a lot of these for post-mortem, so we can look at. But if you look at this across, forgetting about the sample type, but just look at overall, when you look at diagnosis, you get about 21% of these to grow. 15% probably not significantly different out of progressive disease samples. And probably this is because we get larger numbers of tumor cells to work with at diagnosis. But look here what happens when you go to these really late stage patients. So we're selecting in the patient for the ability to grow very well. And these are the samples that killed the patient. So we're particularly fond of these for collecting these to understand what's going on in drug resistance, as well as using these models to find out, and we'll show you some data on that, to find out how we can better treat these patients. Now, the PDXs are basically the same. You're seeing that we can grow a large number of these out of postmortem blood samples. We don't get many to grow out of, because there's not many cells there, out of diagnosis samples. And you get about the same thing you get with cell culture. You get more to growing out of tumor samples in there than you do out of bone marrow. But again, across the board here, not a whole lot of difference between diagnosis and relapse, but you get in a late stage relapse, these postmortem samples, and that's where they really will grow very effectively. And again, these I think are the ones we want to focus on if the idea is not just studying the biology of the cancer itself, but understanding therapy resistance and what kind of drugs will work. Now, relapse cell lines and PDXs, as I mentioned here, higher rate of growth of these, 70% success rate, almost 70% with postmortem samples. These are ideal models for studying therapy and ways to overcome that resistance. They enable studies of molecular mechanisms of the drug resistance that are not feasible due to limited number of tumors patient samples. Go to the COG repository for patient samples and ask how many they've got of relapse samples from patients. Not very many. So growing these things in the mice and in culture gives us the ability to get those samples out to you to study and understand what the molecular changes are that are conferring drug resistance. And establishing these relapse samples paired to cell lines and PDXs from the same patient established pre-therapy provides unique resources for research. And let's look at an example of this. This is some data on some PDXs, one established at diagnosis, and then from the same patient at progressive disease. Those Henry Nguyen published in his MCT papers looking at another drug, but he wanted to compare that to the cyclotopo results. And looking at cyclotopo here, you see that the response diminishes in the survival of the mice remarkably in these, and significantly in these patient-derived xenografts from established here after the patient had relapsed. So something's going on, and we're starting to look at this in depth, but just to look at the first example of this, the shared mutations across these, this is by whole exome sequencing from David Wheeler's lab, really not different. But look at the green here, these unique at diagnosis that are not seen in the relapse patients. So we're deleting these out. And then look at this large number of unique at relapse mutations that are occurring. Not surprising because we're treating these with mutagenic chemotherapy. But large things are going on in these patient samples between diagnosis and relapse. So if you're studying this kind of a patient sample and saying, oh, wow, I've got a new drug and it works, but then you get into the phase 1, phase 2 study with it, it may not work because this is what you're dealing with. And there may be some very shared resistance mechanisms going on. So again, this is why these relapse samples are so important to study if you're looking at trying to develop data to support a new clinical trial. Now let's look at what we have in the repository, the validated and continuous pediatric cell lines and patient-derived xenografts. We've got a number of brain tumors, and they're actually across the whole spectrum. I'll show you those. We have chosen not to try to do PDXs. We get very small samples, and when we can, we cryopreserve these. So if there's somebody that wants to do these, but we decided that one is really beyond the scope of what we can do. So we're not doing PDXs on the brain tumors, we're just doing cell lines. The rest we're attempting to do PDXs on. We've got a reasonable number of Ewings, some lymphomas, some leukemias. We'll show you a little more data on those in a moment. And some retinoblastomas. A large number of these due to Lynn Murphy at CHLA giving me samples when I was there. We've got a large number of these cell lines. Starting to get some PDXs on those. And a reasonable number of rhabdos. Most of these are actually from Peter Houghton that established these. But most of what we've got is these neuroblastomas. And the reason is because the neuroblastoma committee is so in tune with trying to get us samples, and that has worked out extremely well. So we're up to 415 cell lines, 60 patient-derived xenografts. 46 of these we have direct to culture and direct to patient-derived xenograft on. And then we've got 16 of these. One of these pairs is PDXs, the rest are cell lines, where we've got the sample at diagnosis and then from the same patient at relapse. And these are all available to anyone that wants to study them in any of these, but where we have most of the data is in the neuroblastomas, and that's what we're going to focus a lot on showing you. Just to show you some of the spectrum of what is going on in the neuroblastomas, this shows you what they look like, of course, in culture. We have 213 of these that we know came from diagnosis, cell lines. 31 of those are PDXs. We have 120 cell lines from patient-derived cell lines from progressive disease. 13 are PDXs from those patients. And then from post-mortem, we've got 33 cell lines, 16 PDXs. 65 of those have gotten samples. We really don't know what the phase of therapy was. 149 of the lines, and this is a mixture of PXs and cell lines, are MYC amplified, 73 are non-amplified. We got 209 yet to test. 29% of these have out mutations. Most of this data is from IL-MOSE, who ran a lot of these. Some of this is from David Wheeler's lab, where we've got 69 that are known to be wild type and 28 are known to be mutant. And then we have yet to test the rest. There's 335 that we will be testing. In the leukemias and the lymphomas, we've got 13 ALL lines, 27 ALL PDXs. We've got 1 AML PDX, and we've got 3 that came in as leukemia, and we're not sure what they are. 10 of the ALLs are T cell, 9 are pre-B, and 12 we don't know at this point. And in the lymphomas, we've got 4 B cell lymphomas and 2 PDXs, and then 2 T cell lymphoma cell lines and 2 PDXs. And the brain tumors, again, it's only cell lines, and this shows you some of the morphology that we see with these that grow. We've got a spectrum. It's unfortunately limited numbers, but because we get limited number of samples, but we've got anaplastic granulocytoma, a medulloblastoma, an oligodendroglioma, 4 ATRTs, 2 DNETs, 2 GBMs, and then 5 ependymomas, and one of these is a matched pair of pre-therapy, post-therapy. So I'm going to switch to neuroblastoma now only because that's where I've got data that I'm of course most comfortable with as a neuroblastoma person, and I'm going to show you some from IL-MOSE. But what I wanted to do is to just spend a little time showing you how some of these models take a drug out of the laboratory and into the clinic. And I'm going to start with retinoic acid. We used Atra in our early experiments, and this is from a Nature paper I did with collaborators at the NCI. Carol Thiel was the first author on this, and it was, I think, actually the first observation of downregulating oncogene that was reported. And it got us very excited about the potential for this for therapy, but at those days, Atra wasn't available clinically. So we started focusing on 13-cis-retinoic acid, and it's actually fortunate we did that because the pharmacokinetics of this were favorable to what we wanted to do. And we came up with the idea of doing pulse therapy so it would be better tolerated, and took a non-NMYC amplified relapsed neuroblastoma cell line that overexpressed NMYC, and we were able to literally shut it down completely over a long period of time with a couple of pulses of 13-cis-retinoic acid. And this was concomitant with this was a complete downregulation of NMYC protein expression. And this really drove us to a phase 1 trial to see if we could get these levels that we picked arbitrarily. And in fact, in the phase 1 study using this pulse therapy approach, we did get 7 micromolar levels with a 4 micromolar trough. And this really excited us, but what excited us more was when we did this, we got 4 complete responses in bone marrow. And so taking the non-clinical data together with those clinical data, we were able to convince the Children's Cancer Group investigators at that time, Kate Mathey was the chair of this study, to incorporate 13-cis in with bone marrow transplant randomization that we were doing in a quasi-factorial design. And many of you all will know these data that showed that transplant is better than non-transplant, and that the best is the group that gets the myeloablative therapy followed by 13-cis-retinoic acid. And of course, this was extended further by ALSU, then looking at myeloablative therapy and adding on top of the 13-cis-retinoic acid, the dinutuximab, and then most recently in using 2 sequential myeloablative therapies together with this and antibody therapy out the back end. And so this shows you how you can take a cell line, and if you pay attention to the pharmacokinetics, and that's an important aspect of this, is to pay attention to the PK that you're getting in the patients, as well as the concentrations you're using in culture and making sure that these are jiving, that you can take cell line data and get clinical results. But I will say that I think that with the nature of patients being more heavily pretreated these days, I'm not so sure that if we did cell line data these days that it would actually turn into a clinical result. So we and others are focused on using patient-derived xenograft models. And I want to talk about some data from IL-MOSE. This is one of her patients that came from the UK. And this shows you another aspect of this is that as you're going to see, we established a patient-derived xenograft from this patient. And we give the patient's parents the opportunity to name the patient-derived xenograft. And they provided this picture when they named the patient-derived xenograft for their child, Felix. And IL had this patient come see her and treated with crizotinib, and was, when the patient died, was very keen on understanding the resistance to this. And she provided us this sample, and we established a patient-derived xenograft that we provided back to her. And then she did this remarkable set of data looking at, as you can see here, crizotinib alone versus chemotherapy alone versus the combination. And as many of you I'm sure have read the paper, there was a remarkable increase in outcome from combining the crizotinib with the chemotherapy, which has led to the incorporation of this in the current high-risk study that the COG is carrying out. So this is, and what really sold me on these data, these models all showed the same thing. But the fact that you could take a post-mortem derived patient-derived xenograft and see this, a patient that failed therapy with the crizotinib alone, really made me think that this was going to work. And we'll see if it does. That study is ongoing clinically. She extended these data, taking with 2 different patient-derived xenografts, both from post-mortem. These are both from patients that died of disease, so heavily pretreated. We failed them with their therapy. And here she's taken and compared crizotinib to looking at the next generation ALK inhibitor that was developed and showing these remarkably better responses with the next generation ALK inhibitor when these patient-derived xenografts are failing the crizotinib. So I think, again, I am very excited about these data, and in large part because the nature of the models. These are PDXs, and they're coming from patients that failed every therapy we threw at them, including in the case of Felix, the crizotinib. So I think that these kind of data are what, at least myself and I think a lot of other people like to see to move something forward from the laboratory into the clinic. Use the right models, use models not that are easy to treat, but use models that are hard to treat, and show that the new therapy can actually work. Now, we mentioned that the interest in immunotherapy and people using GEMS because of that. And for studying the immuno system, obviously you can't do that in immunocompromised mice totally, but I want to show you some data from Michelle Kyle in my lab that's unpublished, but to show you that you can at least study antibodies. When Dr. Bodie's study showed us that there was likely a really good benefit to adding dinutuximab on top of temozolomide and irinotecan, we got some of the antibody from United Therapeutics and asked a simple question. Paul Sondel and I batted this around, and we thought, well, nude mice have plenty of NK cells, macrophages, and neutrophils. It might work. So we looked to see if we could show something that they weren't able to show in the randomized study because they weren't randomizing with and without dinutuximab. If you take a mouse patient-derived xenograft, and this is a post-mortem patient-derived xenograft, that responds somewhat to temozolomide and irinotecan, but the mice all die from disease, and add antibody on top of it, can this cause a benefit? And that's actually what we saw. And now what we're doing is looking at these patient-derived xenografts that have, as you see down here, the GD2 expression. This is very high in this particular PDX. It's low in these others. And when you go in with the antibody, they have a comparable response to temolorino, but you don't see the addition of the antibody benefiting these particular mice. So this is a non-clinical model where you can study chemoimmunotherapy. And one of the questions that Michelle posed was, well, is this the macrophages, neutrophils, or the NK cells? And she got these R2G2 mice, which are NK deficient. and showed that they responded as well or better to the antibody with no NK cells present. So this clinical result that Modi and others have observed and that we can replicate in the lab is probably driven by macrophages, neutrophils, perhaps by complement. And so I think this shows you that you can use these patient-derived xenografts to not just study chemotherapy, but also to study immunotherapy, at least when you're talking about antibodies and combining them with chemotherapy. Now, I want to point out this particular paper, which is the NCI's PPTC consortium model paper that came out in Cell Reports fairly recently. There's a wealth of genomics data in there that Joe Lin did, is driven by John Maris, and it's a combination of models. We contributed the neuroblastoma models, Richard Locke, the ALL models, Peter Houghton, the rhabdo models. There's brain tumor models in there. There's osteos from Richard Gorlick as well in there, and from Peter. A large number of these models, we don't have any but the neuroblastomas and some of the rhabdos in the repository, but I'm sure that these investigators could provide you these models if you want ones that we don't have in the repository. But this is a very nice paper that shows a lot of genomic characterization of these patient-derived xenografts. And I highly recommend you looking at this when you're considering what models to use in your own studies. David Wheeler did the genomics work on this and did a great job at that. So in summary, establishing these cell lines and PDXs requires viable patient samples to be sent to the repository. And that's something I want to stress. More samples, it means we're going to have more cell lines and PDXs to share with everybody. The cell lines and PDXs are established are freely available to investigators worldwide. This repository is a renewable resource. It's, as you see, has enabled ongoing and future clinical trials. We're very proud of the ability to send these models out and help people do what they need to do. And we really want people using optimal models to enhance their research. So if there's anything we can do to help you with that, please let us know. It's important when you're obtaining data, especially for new agent clinical trials, please consider using mostly models established from patients that have progressive disease, because that's exactly what you're going to see first when you get in the clinic with these in the phase 1 and phase 2 trials. You're not going to see chemo-naive tumors. And finally, none of these models we have would be possible without a group of technicians that I've had over the years, starting with Joanne, Tito, Heather, currently Kristen and Jonas, that establish these models, take very good care of them, and make sure that what they bank there is something that you can use, and then get it out to you when you request it. These people have done a remarkable job, and I really have been fortunate to work with all of them. The collaborators I've had over the years, and I don't have all of them listed here, especially I don't have my students and postdocs that have really done a lot of work on these, but starting with Bob and Kate, John and Niall, and Mike Hogarty, Meredith Irwin, Julie Park, Ro Bagatelle, all of these people have made sure that we get samples and work with us on getting data and samples, and I can't thank them enough. And for you that get models, please realize these are the people that helped you get them. Arlene Arano does gets us the update on the clinical annotation and works with us on the bioinformatics comparison of these. David Wheeler's done the sequencing, and Mike Shear did a lot of the ALO work I showed you. But most importantly, I want to thank the people that we never get to thank, and that's the COG physicians, the nurses, the CRAs, and the parents and patients that make these models possible. And a number of those post-mortem models you see are driven by the parents themselves making sure that happens. And we're so grateful to them for their contributions to this. And of course, nothing could happen without funding, and we have funding for characterizing these from government resources, but especially from ALSF, which has made this repository something that goes on and is able to distribute these models to all of you. So I will end with that and look forward to the questions.
Thank you so much, Pat. It was a great talk, and the repository is an amazing resource. So we hope that people will take advantage of the resource. All right, so we're going to start with questions. This first one here is a multi-part question. It's from Jasmine Lee. And the question is, what are your thoughts on creating PDXs by transplanting a whole tumor chunk versus single cell suspension injections with or without matrigel?
We have not done a lot of that because most of the ones that we grow, neuroblastomas, Ewings, rhabdos, are small round cell tumors. They're very cellular. And also we have a lot of samples that we get that are bone marrow or blood. And so obviously you're not going to do a tumor chunk with that. There's investigators at Baylor that have been doing this with hepatoblastomas, which is where it's incredibly important because by taking those and putting them in there, I think they really get a ability to grow these things in a way that you can't do it any other way. For some tumors, it's probably going to be important, and we certainly are looking at that in some. Certainly with the pancreatic cancers, it's important. But I think that what we're most of what we're growing is stuff that is metastatic, and bone marrow or blood is a common resource for us. So that's why we haven't really done a lot with that. I think it's important to study it, and I think we need to look at comparisons of those for the kinds of tumors where it makes sense. In neuroblastoma, leukemias, lymphomas, I really don't think it makes a lot of sense.
Okay, and then the other follow-up to those are, when the patient sample is collected from the tumor, is it collected from the inner, middle, or outer region of the tumor? And then do you find that the level of O2 they survive in vitro corresponds to the hypoxic or hypoxic normoxic regions of the tumor?
I wish I could say that we have that kind of information on tumor specimens that are provided to us by the COG, but we're getting whatever pathologists out there take and get to us. And so as far as looking at the various regions, if someone's working closely with their surgeon, they could probably do that. We did some studies in the past with some tumors where we tried to look at that, not neuroblastoma, but this was in a sarcoma. And there are some differences across the regions of the tumors, but that's not something that we've been able to do with the samples we get from COG.
So the next comment is, thanks a lot, Dr. Reynolds, for the interesting talk and for maintaining important resources. And this person wants to get some PDXs. And then they followed up with, is there a possibility to get a copy of the talk? So yeah, we will make that available afterwards. Next question from Jamjeet. Is it important to establish immune cell incorporated with the cell line in pediatric sarcoma?
I'm not sure I understand the question. You're saying, is it important for the cells to grow out with other cells in there with them? I guess I'll answer that by saying, when we run human to mouse ratios on these things, as well as look at the histology, they're mostly tumor. And that, for the rhabdos and the Ewings, are most of what we work with on the sarcomas. They're mostly tumor. So I don't think that what's coming in from the patient in terms of immune cells has much of a role here.
Okay. Next question is from Ling Tao, and they said it's a very informative talk. And then could we share a list of cells, PDXs, that are growing in different normoxic and hypoxic conditions? How are cells implanted into the mice to develop the PDX mice models in the repository, IV, subcutaneous, orthotopic routes?
Oh, okay. That's a good question, and I'm sorry, I should have covered that. We basically have done everything subcutaneously. We experimented with some orthotopic and some subrenal capsule. We haven't ruled out using that for some really valuable samples that are low tumor cell content. But we tried intravenous injection versus, and seeing where they grow. We've done a lot of trying those things. And what we've seen is that basically the ones that grow out in subcutaneous are basically the same as what we've seen from the others. Haven't been formally done with genomics or anything like that. Certainly probably worth doing at some point. But also, our goal is to support really translational research and new therapeutic development. And the NCI has focused on using subcutaneous models, and I think for a lot of good reasons. So that's why we focused on establishing those. There may be some nuances in the biology of establishing them in other ways, but we haven't approached that at this time.
So the next question is about the failure rate in PDX grafts for neuroblastoma, which I think you discussed. But the follow-up question is, do you know what happened with those set of tumors that fail, like prior radiation, or is there anything else in common?
I'm sorry, by failure rate, you mean the failure to grow or the failure in the patient?
I think I'm assuming it's the failure to grow, because failure rate in PDX grafts.
Yeah, okay. Yeah, you know, it's a good question. We look at viability of these things going in, and you know, a lot of these are that don't grow are at diagnosis, and there are no prior therapy. There's certainly some that we attempt to grow that have tumor cells and their viability that came out of second look surgeries and or bone marrow aspirates after some chemo, and maybe that influences it. But most of these, what we're looking at is viable tumor cells, and so our assumption, and again, we don't have any way of knowing this for sure, but our assumption is that what we're looking at is an intrinsic inability to grow in the conditions we're putting them in. They're clearly growing in the patient, so that leaves open finding the conditions that will grow them, whether it's in a mouse or in the tissue culture.
Thanks, Pat. It looks like that is our last question. Unless anybody else has another question, they can get it in right now. But again, Pat, what could you just go over one more time what people would do if they wanted to get some samples from you?
Sure. First of all, I'll comment that in case there's any industry people looking, we have been working with the COG to get a revised industry MTA. That's a long story, and I'm not going to go into a long story, but it hasn't quite happened, but I'm hoping it'll happen soon so that we can get these out to industry without some of the questions that and concerns they had. There were some issues with the MTA. That said, in doing that, we're hoping to have a very simplified one single MTA for everybody. Again, it still hasn't happened. So in the meantime, we have two MTAs. One, they're divided into open distribution list and closed distribution list. And the closed distribution is really just those that we have that haven't been published, haven't gotten out there, that with multiple people may be working on, and somebody's trying to characterize and get a paper out. So we're just trying to limit what people do with them a little bit, just to preserve somebody who's taken them on to characterize them. There's not a lot of that going on right now. So most of what we're talking about is that open distribution MTA. It's a COG MTA. You can download it off the website. You basically work with us to decide what it is you want and need. And I say that you can look at the list, and there'll be larger lists going on the website soon, and see if there's ones there that you know you need, and just put them on there. But if you're trying to study something, contact us, and we'll have a dialogue back and forth with you and say, here's the kind of cells we think that are the cell lines or PDXs that'll serve your needs the best. And then we could just add those to the MTA. Your people sign off on it. We send it through to the COG. They sign off on it fairly quickly. You give us a FedEx number, and we send you the models along with the data sheet that tells you a little bit about them. And certainly we're there to ask questions of and help you get them growing.
Great. And then what if somebody had samples they thought would be good candidates for you to have in the repository? Are you ever looking for more samples?
Yeah, we're happy to bring ones in. We've done that before on some samples, some limited numbers. The thing is, we have a commitment that we're only going to have something in there that we know what it is, and so if we bring something in, we'd prefer to have some original patient material, either an STR or DNA, to be able to validate that. But if we can't do that, we'll run an STR and make sure it's unique in the database, that it's not, in other words, a cross-contamination. And then we just need whatever paperwork that the institution that's coming from is comfortable with, so that we can use it. We need to validate that it came with consent, and then we'll run it for mycoplasma, and if it's a PDX, we of course run it for human pathogens, and then we can expand and incorporate it into the repository. We'd leave the name as it is, whatever it comes in, and of course in the repository we'd note that it came from whoever submitted it.
Okay, and then one more question just came in. The question is, do you know where someone can get the genomics and functional data on the models in the cccells.org?
Okay, a great question and something I should have also covered. So for the data set that's in that article that I showed you, the PPTC data set, that is on dbGaP. And I believe you have to contact John Maris. I hope I'm not sending too many people to John on that, but I think that he's the contact person to get that released, and then you can get permission to go through dbGaP and get that data. We have a larger number of RNA-seq data available, and it was decided that we're not going to just put it out there until we get it actually peer-reviewed. But if somebody has a particular gene they're looking for or anything, we're happy to look through it and say, here's the models that express what you're looking for. And we're hoping to get that data in the literature soon. And then we're going to make that available through dbGaP, and it will be less restricted, I think, than the other stuff. We're going to hopefully make it as open as possible.
All right, well, that brings us to the end of the questions, Pat. And I just want to thank you again for giving this talk today and for everything you do to keep this repository going. I know it's been a passion of yours for quite a while, and we appreciate the work.
Well, Jay, I appreciate you and ALSF, because it is a passion of mine, and the repository wouldn't exist and be available to people without what you guys do. So thank you.
Well, it's our way to sort of give back and help the research community as a whole. So we're glad that we can do it, and keep up the great work and stay safe, and we'll talk to you soon, Pat.
Thanks. Same to you, and same to everybody out there.
All right, take care.